Okay, this is the second class of chemical biology, and last time we talked about the size of the molecule starting from the carbon and carbon, and then we talk about amino acid and protein, and also we calculate about how big they are. For the example, lysozymes and albumin. Lysozyme is about 200 something amino acid. Albumin is about 600 amino acid. The first one is XYZ in scale about 2.4 nanometers. The second one is about 3 point something nanometers. DNA is the most important biomolecule, which is the protein and DNA, or RNA is the most important biomolecules, I should say. So today we talk about how big they are. So at the end of this class, we will combine the knowledge about the DNA or RNA plus the protein, so you can combine those, what is the relationship between those things. So the DNA, the basic structure is a sugar. So I want to recall your memory about 5 member rings of the sugar, and then you have secondary hydroxyl groups, and you have 5 members, the hydroxyl groups, to which you have the phosphate group connecting to here. Then you have the phosphate groups are looking like this structures, and then to here we have another. The unit comes in here, and also the below it you have another structures which is phosphate, also comes through this called phosphodiester, which is phosphodiester in this structures. And then of course you have another 5 member rings are coming actually in this way. You have like this. So you can repeat it this kind of things. This is the basic backbones of DNA, and then you have AT and GC, which is base pairing. I'm not going to focus on this two, because we want to know the overall size of the DNA, the length, which is the size of the DNA. And then to which this is a base, or either ATGC comes in here, binding with a hydrogen bonding. In the case of the A, you have a T, and then the same phosphodiester and 5 member ring sugar backbones comes in here. So this is a double helix of the DNA structures. This is the knowledge that you learned in your biochemistry class in the maybe 2 years ago, 3 years ago. Today we have focused on a little different thing, which is the size. That we start from this phosphate group, and then as a similar, we have last time. We calculate the unit of the protein, which is amino acid, and then we multiply this the size of the amino acid. Similarly, we have, say, the unit we can determine from here, and then maybe from here. This is the basic unit of the DNA in the backbone, and then the second unit is going to be here. The third unit going to be here. So this is the base pairing. So we have one base pair, means this is one base pair size. This is a 2 base pair size. So typically our gene is about 300 or 600,000 gene base pairing that is coding the one protein. So this is very small part of the DNA that coding into the protein. So by the way, we have, say, from here to here, the number of bonds are, you can simply count it 1, 2, 3, 4, 5, 6. So you have a 6 bonding, which is about 6 angstrom. Again, last time we talked about this is maybe 1 point something angstrom. This is 1.4 angstrom, 1.2 angstrom. But you cannot actually add those things, because you have to considering about the angle between these old atoms. In the case of the sp2 or sp3 orbitals. So this is a 6 angstrom. So now, if you have, say, connecting to before the first class, which is lysozyme, that is about 200. It's about 260 the amino acid. Then the DNA coding required to encoding this 600 amino acids, one amino acid coding is, we have a 3 bases are required, right? Such as ATG encoding glycine, TTG encoding the lysine, CGC encoding to the tryptophan, something like this. You need 3 base pairs to encoding the one amino acid, right? So then if you need fully encoding 216 amino acid, so simply you multiply by 260 now, then you have about 780 bases, right? So the 780 bases, now this is one base pairing length, okay? So then now we can multiply by 6 angstrom, okay? So then now we can calculate this, the overall length, which is 780 times 6 angstrom, which is 680 angstrom. So if you recalculate nanometers, then 468 nanometers, which is about 0.5 micrometers. So which is very big, very, very big. Even the length, of course the thickness is very small, but the length, if you're fully stranded to code the simple lysozyme protein, the DNA required the length is about 0.5 micron, which is very big. That's why people said, if you have a single cell, you take out all the DNA in our cell and connecting into all the in the line by line, then the length goes to the moon, right? So that's why it's very, very long, and then such a long DNA of the length is well packed in a single cell, even in a nucleus. So this really big length. So you can imagine now, lysozyme is 260, about that number of amino acid. So then the albumin, which is about 3 times more in number of the amino acid, right? 600 more something amino acid. So that means it's already 1.5 micrometers in DNA length, which is more than 1 micrometer we can see, actually, see the length. So this is very big molecule, I should say, and we have to think about the DNA in the insides. So that's why if we recall the memories that you have a DNA duplicated DNA structures in here, and then we learn about, say, when DNA becomes RNA, RNA becomes a protein. There's a schematic photos that in biochemistry class that proteins are coming into here, right? Then DNAs are such a long molecule, and then from here, this protein, which is we call the transcription factor. Transcription factor means that the protein that controlling to make this DNA into the gene, into the protein, so that this transcription factors bind into such a specific DNA recognizing sequence, and then they start to control all the things that they're recruiting all the proteins that another pathways of the protein producing machinery processes begin. So that's why you think about it. I mean, the first class about 2 point something nanometers to 4 nanometers in here. And then this is kind of sphere or cubic shape. And the DNA is such a long molecules that in case of the lysozyme, 0.500 nanometers required. So this DNA from here is actually have a multiply 100 times of the DNA. These proteins are controlling this, moving through back and forth in this DNA chain. So that's why the DNA is such a long molecule. That length has to be controlled like a protein. Also, protein is a long molecule, but they are folded like a secondary structure, alpha helix, beta strand. And then they have hydrophobic interactions between lipid-like amino acids. They can make a well-packed cubic or sphere-like structures. Same as DNA. Same you have, say, in here we have such a long DNA molecules called histone. The histone is about, say, if you think about histone similar to lysozyme size, then this is histone. Then it's about 2.5 nanometers, you can imagine. And then this histone and surface charge is completely opposite. So DNA is its backbone, it's phosphate, oxygen phosphate. So you have an O minus group, which is negatively charged in all the backbones throughout the DNA chain. So that you make a binding between the DNA, the histone has a positive charge. And then DNA here, we have all the, because of the phosphate group, you have all the highly, highly dense, negatively charged along this backbone, which is this negative charge individual charge is coming from individual phosphate group that we described before. So when histones come, and then they have interactions in here, and they make a folding. So this DNA goes in here, maybe wrapping around to the histone, because there's positive charge also here, that comes, all the negative charge, this DNA comes from this strand to fold it. So then the histone, another histone comes in here, they make a folding of the DNA. So they start the packing of DNA, such as 0.5 micron, such a long DNA, then I now well pack it to put into the small nucleus. So this is kind of scales that we talk about. So that's why proteins are such a small, but DNA is small but in the very, very long in length. So that his proteins such as histone comes and they wrap around. Another histone comes and wrapping around and make all the DNA into this packing, well packing into the small space. So this now providing you an idea about this virus. This virus is such an amazing structure, since virus itself is not a living thing. So we have a virus so virus is not a living thing. It's not a living organism. Maybe there will be another classes we talk about the viruses. But itself, and simply speaking, the virus is not an organism. It's not living. The living organism defines that when have a cells, then cells can divide it. 1 becomes 2, 2 becomes 4, 4 becomes 8. That is a cell. There's a living thing. A virus cannot amplify themselves. Virus need a cell. And the virus can come and then go into the cell. They use their cell's machinery. The cell help the virus to amplify. The amplified virus kill the cell and it goes out. So this is a virus required. So that's why the cell becomes, in this case, becomes called host. So the virus becomes a guest, cell becomes a host. So virus is not a living thing unless they're floating around in the air. Then becomes living things only when the virus comes into the cell. Okay? So that is the difference between the bacteria. So bacteria, such as E. coli, we know we have E. coli in our body. So bacteria is indeed a living thing. It's a living organism because the one bacteria in E. coli can divide it in our body. So in our everywhere, bacteria is everywhere. They can divide it, and then also they build their home using the polysaccharides we talk about later. Then they build a home and a house in which the bacteria is living there, and they divide it. So you have infected by the bacteria, so you get sick. That means they goes into the bacteria. They build their own house. They divide it into your body, and then there's a certain population. Over a certain population, you see a feeling of the sick because now the bacteria is feeling very safe. They secreting some the molecule from this bacteria that's molecule that are harmful to your body. So there is kind of mechanism how they making you sick. So the virus now. So I'm not talking about the bacteria here. So virus is a guest, and also cell is a host, right? And the virus has their gene, which is some certain can be type of the virus can be they have a DNA inside, and also certain virus they have an RNA inside. right? The DNA RNA is simple only when you have the backbone, you have this. In here, I will recall your basic structures, and then DNA has the no oxygen, and then RNA has the oxygen. Only that there's a difference, right? So anyway, basically same structures. And then the DNA packet or RNA packet virus, so we think about how big they are. So what's the size of it, right? So probably you can imagine now, because the virus has inside the DNA and RNA, so their house, which is the surface, the packing materials is basically protein. So they have structures like DNA or RNA is packed by the protein. It's like this. It's not a two-dimensional thing, it's in the spheres, like a three-dimensional thing, it's packed. Of course, there's a certain, the coronavirus, for example, they have a spike proteins, looks like this. Another proteins that comes in out of the hair, so this is mediating by the cell in your respiratory tract, and then it goes inside. So this now, our topic is talking about the size. Now imagine that roughly we talked about the last time 200 amino acid in size of the protein, 600 amino acid inside the protein. But the amino acid number totally different, but their size is not a big difference, because 2 point something nanometers to 3 point something nanometers, only 1 nanometer is different. So we can imagine now, we can estimate it that if this is the 3 nanometers in protein, if this individual proteins has 3 nanometer in size, then your question is 3 nanometers are size is in the other part, right? So you have the virus size, it has to be bigger than 6 nanometers in size. But should be much, much larger, because the DNA or RNA there, which is an essential part of the virus, it has to be inside. So then you have to know how long DNA or RNA exists inside this virus, right? So that is the kind of key things. So typically, the size of DNA, for example, we talk about, say, not the virus, the coronavirus, and typically very useful virus for the gene therapy. We're developing a global pharmaceutical company use the virus, which is useful virus for the gene delivery, which is called adeno-associated virus. I will talk about this virus, for example, to estimate the size. So this is called AAV. The associated means that actually people found adenovirus first, but they found that adenovirus has like this big, and then there's always another, like a daughter-like a virus attached to it. So this is adenovirus. So people is wondering that what is this, and then eventually found out that this is also virus. But the virus is always come with the adenovirus, so that's why they call it adeno-associated virus. So we have adeno-associated virus. Probably in the worldwide population, more than 60% people has already adeno-associated virus in our body. So this is not harmful. That's why people use this virus for a vector to incorporate their useful gene and deliver our body. So anyway, this adeno-associated virus, which is AAV, has about 6 or 7 kilobases in DNA size. This is a DNA virus, not an RNA virus, like a coronavirus. 700 kilobases means that 7,000 bases. Remember that one base requires what length? In 6 angstrom. So one base is 6 angstrom. So we have a 7,000 times 6 angstrom. So that when you imagine this, all 7 kilobases DNA are fully stretched by our hand, using our hand, instead of not unpacking by the histone, like things. Then 2, right? And then 0, angstrom. We calculate the nanometers. Then 4,200 nanometer if you need. micron, which is more realistic unit, then you have 4.2 micrometer, which is really big in length. So 4.2 microns. 4.2 microns, your hair thickness, not a length, in thickness, its hair is about 10 micron. So it's about half size of in diameters in our hair, typical hair. So very big length. So that cannot be packed into this such a small virus, right? So they have also mechanism that like a histone-like mechanism, not actually same as a histone-like mechanisms, but they have a packet with like this DNA or packing like this very well goes into this virus. So this virus, for example, has their own mechanisms like similar to the histone. Then they have packing, and then this eventually you cannot imagine how they big are. So it can 22 or 23 nanometers in size. So now we can think about then if the DNA in such a 4.2 micrometers in length is able to pack into the 22 or 23 nanometers in size of the virus in the empty spaces inside, then how well they pack it to here. So you can imagine. So you can think about this is kind of homework that you can think about. I mean, this is what a DNA bases from how many bases from here to here are, say, 10 amino acid, 10 base pairs or 20 base pairs, something you can think. So you can, one base pair is 6 angstrom, 10, 6, 60. So about 16 angstrom means 6.6 nanometers. So you have at most you have 20 base pairs happen here, already occupying here. So 20 base pairs that folded 20 base pair, 25 base pair, 20 base pair folded here, then making all 7,000 base pairs is inside. So such as amazingly well packed mechanisms as viruses they have. So now this virus, adeno-associated virus, AAV virus, is about 22 nanometers. The cells is about 10, 20 micrometers. So think about, say, if you pick this number, for example, in the cell, which is host, which is 20 micrometer, it's about 20 nanometers, so 1,000 times the difference. So 1,000 times difference means you have 1,000 times bigger in the length. The volume is much larger. Volume is another line. You have 1,000 times. If this is 1,000, then you have one out of the 1,000 particles that comes. They goes into the cells. That is, you can imagine how small the particles are that goes into the cell. So this virus goes in, and then they can divide it by themselves using their machinery of this cell, and then they can make a number of viruses goes in there, and then they eventually come back from out of the cell, and these individual particles of the virus, which is as soon as they are out of the cell, they are not a living organism. They're just particle. And then they spread it, and then it goes into a different cell. They multiply using the host mechanism, and then they multiply again, so it becomes sick. Okay, this is the end of the second class, and then we next time talk about another size in molecule, which is polysaccharide. And polysaccharide becomes hydrogel, and hydrogels the pore size, and through which we have a protein and DNA comes in. So we learn about the electrophoresis, that DNA electrophoresis, protein electrophoresis. They can talk about also in size classes. Thank you very much.